a wild-type FLT3 at the time of diagnosis. This was a next iteration of sorafenib plus chemotherapy, but this was only in patients with ITD AML. Similar idea, 7 and 3. They pushed the sorafenib back a little bit into the treatments, very similar to the way in which it's given by the Ratify group with midostaurin. Then they gave maintenance therapy with sorafenib or placebo for 12 months. Remission rates were on par with what we discussed earlier, 60 to 59%, placebo versus not. Transplantation rates were not statistically different. What was statistically different was an event-free survival benefit, favoring the sorafenib arm, but it was exactly 12 months. And the 3-year overall survival was slightly higher in the sorafenib arm, but it wasn't statistically significant. Overall survival in this final curve here does show you that there's no real difference amongst the group. So this is an interesting study telling us a little bit that we haven't reached where we want to be with FLT3 ITD and sorafenib yet, so we need to try something different. And as they were doing the sorafenib study, the group, Richard Stone and his colleagues were accruing to this study, which was a similar idea. Cytarabine, daunorubicin, midostaurin, 50 milligrams twice a day, day 8 through 21, or placebo. But it was a FLT3 mutated patient population, either ITD mutants or tyrosine kinase domain mutants. Then patients were consolidated as we typically do, also given midostaurin in consolidation, and then a maintenance 12-month treatment with placebo or midostaurin. And the results of this study were reported in the New England Journal earlier this year, showing remission rates that weren't statistically different between the midostaurin arm and placebo arm, maybe slightly higher in the midostaurin arm. But what was different was a marked improvement in overall survival, 74 months versus 25 months in the placebo arm. Transplantation was slightly higher in the midostaurin arm. 4-year overall survival, again, was slightly higher in the midostaurin arm. Looking at the subsets of disease here, broken down on tyrosine kinase domain mutations versus ITD mutations, the tyrosine kinase domain mutations appeared to have the highest benefit from receiving midostaurin over placebo, although the confidence intervals are wide here because the number of patients is significantly lower. But there didn't appear to be any difference between patients who had a high allelic ratio of FLT3 versus what they considered a low allelic ratio of FLT3. Hazard ratio is very similar. So this is become a standard approach for us for FLT3 ITD and tyrosine kinase domain mutations, as this is really the first study that's shown that we have an overall survival benefit by adding a tyrosine kinase inhibitor into the treatments. And the other thing that I think we learned from this study, which I think was interesting and was discussed briefly by one of your colleagues, was that there may be now real data to justify transplanting patients with FLT3 mutated leukemia in complete remission 1. And in the appendix to the study that was published, this was one of the figures that they presented, and I'll walk you through it briefly. Patients in the top 2 curves here were either patients who had midostaurin and a stem cell transplant in first remission, or placebo and stem cell transplant in first remission. And as you can see here, those 2 curves would suggest that taking the inhibitor plus a transplant in first remission showed some benefit over time. Whereas midostaurin didn't confer any degree of benefit if you waited for relapse and then transplanted patients at that point, which is not not surprising, but it is, in my opinion, justification to do whatever we can to prevent patients from getting on the transplant or trying to get a transplant in second remission. So I think we've reached a point now where we've have to figure out where and how to apply these drugs in particular patients, because it's clear that there is some differences in FLT3 mutant leukemias that have influenced how those leukemias respond to treatments. Patients either can have what we call heterozygous mutations, meaning one chromosome 13 has the mutation versus both chromosome 13s had the mutation. Copy number and expression of that allele can be influenced by epigenetics. How potent these drugs are and how selective they are for leukemia. And clinical status of the patients, either newly diagnosed or relapsed, can influence a number of things, including how much FLT3 mutation is there. And lastly, whether the patients at any point are known to have a kinase domain mutation. That, in my opinion, really helps me pick which drug to use. And a few years ago, this was a really interesting study of the clonal evolution of cancer genetics in leukemia cells. And really shouldn't focus so much on the genes here, but the colors of this figure. And what it represents is at the time of diagnosis, there are a number of specific clones of disease. And in this cartoon, the purple clone was the largest. But despite the fact that that was the largest clone at the diagnosis assessment, it wasn't the one that relapsed. In this figure, it was the yellow clone, which was one of the smaller clones. And it's just an illustration that I put up to just tell you that just because what you start with doesn't necessarily mean that's always what you have to target in the end. And the FLT3 is often one of the drivers of these late relapses, even if it's in a small subset of disease at the time of diagnosis. And the way we grade that, or the way we gauge it, is something called allelic ratio. And what that means is it's a measure of the amount of FLT3 ITD found in a DNA sample. When we take the DNA and isolate it from the leukemia, they'll do PCR to look for these mutations. Wild type FLT3 runs a certain length. We know how long that is. When patients have an ITD mutation, it lengthens the length of the PCR product. That takes longer to run down the gel. So you get a band that is above the control band. And that's a patient who has a FLT3 ITD. And the relative amount of these bands, this higher band versus the lower band, gives you the ratio. So there's two examples up here. One is a low ratio patient, the other is a high ratio patient. High ratio means that most of what the PCR product found or replicated was a larger fragment of FLT3, meaning that there was an insertion there, and as such, this is an example of a high ratio patient. I think it's important to just think about this because it does get confusing when this is talked about a lot. Anyway, high ratio refers to often patients who've lost heterozygosity in the chromosome, and that occurs as a clonal evolution occurs. Several years ago, we tried to sort out whether allelic burden could be useful in determining which FLT3 inhibitor to give to patients. And this is a real busy slide, so I'm going to walk through it briefly. we separated leukemia samples into high ratio samples or low ratio samples. And I just, in this figure, I want you to focus on the dark lines versus the light lines. And this is a example of selective inhibitors. AC220, sorafenib, sunitinib were inhibitors that didn't have a lot of off-target effects, meaning they only hit a few kinases, whereas other drugs like midostaurin and listartinib hit a lot of kinases and are what we consider pan-kinase inhibitors. So when we exposed leukemia samples to these narrow index inhibitors, they really only did a good job at inhibiting high ratio FLT3. This lower slide means that they're inhibiting it better. And when we compared a broad kinase inhibitor, and this figure was lisartinib, versus AC220, which is a narrow inhibitor, the broad inhibitors could inhibit both high and low leukemias quite well, whereas AC220 was only able to inhibit the high ratio samples. So this in our mind argued that the better drugs for patients who have a low allelic ratio are these broader kinase inhibitors, as they're able to pick off more of the leukemia clones and prevent them from recurring. And we went further to examine this by dividing up the samples into diagnostic versus